Fantastic. So, hello everyone. Thank you. Um, Welcome to this month's Cancer Genomics Cloud webinar. My name is Dave Robertson from the CGC team at Valsera, and I'll be your host for today. We're excited to have Dr. Sonam Tulsyan. She's a visiting postdoctoral research fellow at the National Cancer Institute Department, DCEG. She's joining us to share her recent work on HPV positive head and neck cancers. So in her talk, Dr. Tulsyan will walk us through how she used long-read whole genome sequencing to analyze tumor and normal tissue biopsies. Her study sheds light on how HPV integrates into the human genome and what that means for cancer development and potentially treatment. So we'll have time for Q&A at the end, so feel free, if you think of a question, feel free to type it into the chat or the Q&A box that we have on Teams, and I'll help guide that discussion after the presentation. So with that, please join us in welcoming Dr. Tulsyan.
Thank you, Dave, for a nice introduction. As you told me, I am a postdoc fellow in Dr. Mike Dean's Laboratory of Translational Genomics in NCI. Today, I'm going to talk about molecular analysis of HPV-positive head and neck cancer tissue biopsies using whole genome long-read sequencing. Giving a brief introduction of the cancer, head and neck squamous cell carcinoma is the 6th most common cancer in the world. Oropharyngeal squamous cell carcinoma, which is a type of head and neck squamous cell carcinoma, have an age-standardized incidence rate of 2.5 per 100,000, which is one of the highest in North America across continents. Several risk factors are associated with OPSCC etiopathogenesis. They include tobacco, whether in the form of smoke or chew, alcohol, human papillomavirus infection, inadequate nutrition, and poor oral hygiene. Persistent HPV infection increases in risk of OPSCC, which is responsible for nearly 70% of all OPSC cases found in North America. Comprehensive understanding of the complex molecular events within the HPV plus tumor remains a critical knowledge gap. Therefore, we thought to long-read whole genome sequence 16 OPSCC fresh frozen tumor non-tumor biopsies. These samples were received from University of Maryland. A brief introduction of the HPV genome. Papillomaviruses are small, non-enveloped DNA viruses that have a diameter of 52 to 55 nanometers. These viral particles, they contain a single double-stranded DNA molecule of 8,000 base pairs, which is bound to cellular histones, and these are contained in a protein capsid composed of 72 pentameric capsomers. They have 8 genes, 6 early genes and 2 late genes. The early genes are responsible for replication and transcription, whereas the 3 early genes, E6, E7, and E5, they act as viral oncogenes. The late genes, they are involved in capsid protein formation. It also has a URR, which is the untranslated region or the local control region, which has the transcription start site, various transcription factor binding sites. Once the HPV infects the normal cell, the cell gets infected. In 90% of the cases, the immune system successfully clears the HPV infection on their own within 2 years. Persistent HPV infection, HPV genome can either stay in its episomal form or in the integrated form in the host. This leads to pre-cancer stage, and this persistent infection causes genomic instability, loss of tumor suppressor genes, activation of oncogenes, which finally leads to cancer. When the HPV infects the host chromosome, it can either stay in its episomal form or in its integrated form. This episomal form can be a monomer, that means a circular 8 kb genome, or it can rearrange itself into multimers. When this monomer form of HPV integrates into the host chromosome, it leads to type 1 integration. That means only partial HPV genome copy is inserted into the host chromosome. And when the rearranged multimer form integrates into the host chromosome, it leads to type 2 integration. That means more than one HPV copy, more than one HPV genome copy is inserted into the host chromosome. These integrations can be simple or complex. Simple if it has integrated into the human chromosome, which has with two breakpoints, but if it has integrated into several places with more than two breakpoints, it leads to complex integration. These integration, they disrupt the E1 and E2 genes. E1 and E2 genes are responsible for viral DNA replication and gene regulation, which leads to increased expression of viral oncogenes like E6 and E7. These viral oncogenes, they degrade the tumor suppressor proteins like P53 and PRB, which are implicated in cancer development. This is the slide which shows the flow chart of my study. So as I told you before, also we collected 16 samples. These samples were collected from University of Maryland. Out of these 16 samples, 6 samples were paired, like the tissues were taken from tumor and non-tumor histopathologically confirmed OPSCC cases. DNA was extracted and quantified. Library was prepared by Oxford Nanopore LSK114 chemistry. These were loaded onto the ProMethION flow cells on P2 solo device, and data in the form of FASTQ-Pass files was collected after 72 to 100 hours of the run. All the bioinformatic analysis was done either on CGC or Epitomy Labs. FASTQ-Pass files were aligned to CGC to get the BAM files using the alignment tool on Cancer Genomics client. Precise mapping of the integration sites were done using IGV. Detection of HPV sublineages was done in software. The sample's ancestry was checked and confirmed by Ancestry tool on Cancer Genomics Cloud. Copy number variation plots were created using R scripts on Data Studio on CGC. SNPs and the somatic variations were called out from Epitomy Labs, which is the software for analyzing Oxford Nanopore technology sequencing runs. But these were annotated, the SVs were annotated through Anode SV tool on Cancer Genomics Cloud. This is the table showing the demographic details of all the OPSCC cases included in the study. There were 10 samples included in the study. Out of the 10, the 6 were paired, like we have the tumor and the normal tissue of these samples. All 9 out of 10 samples were male and 1 was female. We did not have the clear demographic details of the 2 patients, that is patient with the ID 2 and 5, but the gender was confirmed through whole genome sequencing. 7 out of 8 patients were above 55 years of age, and only 1 patient was below 55 years of age. 3 out of 10 patients had the African American race, which was confirmed by the Ancestry tool on Cancer Genomics Cloud, and 7 out of 10 has the Caucasian ancestry. All samples recruited in the study were non-Hispanic, and none of them was present smoker. As I told you before also, sequencing was done through Oxford Nanopore technology. LSK114 chemistry was used to prepare the libraries. Libraries were checked on Flongle, and they were deep sequenced on ProMethIONs. Data was collected and analyzed on several tools like Epitomy Labs, Cancer Genomics Cloud, IGV, Mega 11, MitoMaster, BLAST, and BLAT. We made sure that we achieve a sequencing coverage of 30 to 40x for tumor cases and 10 to 15x for normal samples. So all the FASTQ-Pass files which were generated from the sequencing run, they were uploaded to CGC. They were aligned to reference genomes like HPV and human genome on alignment tool on CGC. The aligned BAM files were viewed on IGB to map the integration breakpoints. Out of the 10 patients, we noticed 9 of them are HPV-16 positive, while 1 is HPV negative. Out of these 9, we found in one patient that only one read was aligned to HPV-16 genome, showing that it's weakly positive. So we only proceeded ahead with the analysis of the 8 samples. Out of 8 samples, we found 6 to have episome HPV pattern and 2 to have integrated pattern. The frequency of episomal was very high in our studied OPSCC cases when compared to integrated pattern. This is the IGV screenshot of the 3 more HPV-16 episomal pattern cases. So episomal means that it has a full circular 8 kb genome, and these are the 3 BAM files showing the episomal HPV-16 pattern. No reads were aligned to human chromosome. We found one sample to have rearranged multimer in HPV-16, and this rearrangement was from 7557 to 3330. So these are the read coordinates in HPV. We found that after 7,557 base pair, the next read was 3,330, confirming the rearrangement. And when we looked at the predictive transcription factor binding sites in HPV-16, local control region or untranslated region, we found that there is a duplication of these transcription factor binding sites, E2 transcription factor binding sites, which are just before the data, which is the transcription start site. I'm saying duplication because you can see the region which is highlighted in black is the URR or the local control region, and the rearrangement involves more than one copy of LCR region. We noticed that one sample, episomal sample, has a deletion of 636 base pair from 4892 to 5528, and this was clearly visible in the IGV BAM file. We also noticed that one episomal HPV-16 has an insertion of 257 bases, which we noticed it here and found that this region, which was duplicated, was in the local control region from 7310 to 7567 base pair. Again, we saw this in the transcription factor binding sites in HPV-16 genome, and we find when there is a duplication or an extra region of this, so the transcription factor binding sites, they are increased, therefore leading to more transcription and uncontrolled cell division. We found 2 samples to have integration. Out of those 2 samples, one had type 1 integration and one showed type 2 integration. Type 1 integration was found at chromosome 11q. The purple arrows highlight the region, the breakpoint junctions, and the map also shows that the chromosome 11 integration took place at 6,150 base HPV genome, and the right junction is at 2,985 bases. We confirmed our observation on the EGI38 BAM file, and we found that HPV integrates into this intronic region of the PLAT5 gene. This PLAT5 gene is responsible, encodes enzymes which are responsible for lipid metabolism. It encodes phospholipase A12 activity enzyme and acyl transferase activity, which catalyzes fatty acid release from glycophospholipids and transfers acyl group to phosphatidyl ethanoalamine and forms N-acyl phosphatidyl ethanolamine, which is a precursor for lipid metabolism. Several studies have shown that that this gene acts as a prognostic marker in glioma. Its higher expression is correlated with worse clinical outcomes, and it is also found to be overexpressed in breast cancer cell lines and ovarian cancer cell lines, which suggests that it has a role in tumor progression. Another sample which we found to be integrated was patient with ID8, and it has type 2 integration. Type 2 integration, that means it has more than one genome copy, more than one HPV genome copy, and it was found to be integrated at chromosome 14q. This was again confirmed by viewing the AG38 BAM files on IGV, and we found that the left junction, at the left junction, HPV integrates into the intronic region of MPOL1 gene, and then the 3' UTR region of FOXA1 gene at the right junction. When I looked into the functional relevance of these genes, I found that MPOL1 gene was originally characterized in developmental disorders like mirror image polydactyly, and studies involving chromosome 14 disruptions or rearrangements have identified this gene as a tumor suppressor gene in nasopharyngeal carcinoma. Functional studies have also shown that restoring MIPOL1 expression in nasopharyngeal cancer cells suppresses growth, supporting its role as a tumor suppressor. Therefore, loss of this gene or mutations in this gene may drive certain cancers and may warrant further research for biomolecular potential, because I only found studies related to nasopharyngeal carcinoma for this gene. The other gene where HPV was integrating was the FOXA1 gene. It acts as a pioneer transcription factor, enabling estrogen receptor and androgen receptor binding in hormone-dependent cancers. Studies have shown that it is favorable in estrogen receptor-positive breast cancer, as its high expression leads to better prognosis and therapy response. It is also found to be oncogenic in studies have shown that it is also found to be oncogenic in colorectal and lung cancer. In small cell lung cancer, it acts as an oncogene, activates HER2 transcription and PI3K-AKT pathway, promoting proliferation and poor prognosis. Therefore, this gene has a dual role. It can be oncogenic or tumor suppressing depending on the tissue type, and it also acts as a predictive marker in estrogen positive breast cancer for better outcome and therapy responsive. Therefore, it can act as a potential target in cancer where it may drive disease progression. This gene integration was observed in the only female OPSCC case of the study. We also checked the HPV sublineages and we found that A1 sublineage is the most common among others which we found in our study. A1 and A2 these are the most common HPV-16 sublineages which are found in United States and Canada, whereas D2 and D3 are rare in American population, and they are most frequently found in Central America, and they are found to be associated with cervical adenocarcinoma cases of cervical cancer. We also predicted the haplotype by using the MitoMap software, and we found that 7 of our 10 samples have the Caucasian ancestry, and this was confirmed by the predicted haplotype H, T, and I. 3 out of 10 samples have the African ancestry, which was confirmed by the L1 and L2 haplotype group. Again, we did the ancestry was confirmed by the ancestry tool on CGC, and we found these 3 patients to have African ancestry. However, these 7 patients have European ancestry. Also, The tumor and the normal haplotype was predicted for normal and the tumor cases, and they predicted they have the same haplotype, confirming that the samples came from the same patient. This is the slide showing the summary of all the cases studied. As I told you before, also 8 out of 10 were found to be HPV-16 positive. We observed more A1 and A2 sublineages in our sample. 7 out of 10 samples have the European ancestry, and 6 out of 8 studied samples have HPV episomal pattern. We also we also analyzed the TCGA HPV positive head and neck cancer data, and we found that most of the samples are HPV-16 positive, and these samples have more integration status when compared to episomal status. So therefore, HPV-16 was found to be the most integrated, was found to be integrated into the host genome at a frequency which was approximately twice of that of episomal HPV-16. We checked the distribution of HPV-16 sublineages in TCG HPV positive head and neck cancer data, and we found that the most frequent sublineage was the HPV-16A, and HPV-16A, out of this HPV-16A, HPV-16A1 was the most frequent one, and this is the chart which shows the distribution of HPV-16 sublineages according to HPV pattern. We also stratified the TCG HPV positive head and neck cancer tumor stage data with HPV patterns, and we found the integrated HPV pattern was 3 times more common in stage 3 and stage 4 tumor cases. Unfortunately, presently we don't have the clinical stage data for our patients presently, and I couldn't correlate with our HPV, the HPV patterns which we noticed in our study with the tumor stage data. Coming back to my own results, we also did we used the human variation workflow on epitomy labs to rule out those copy number variants, single nucleotide polymorphisms, and structural variants. And this is the image which shows the whole genome distribution of copy number variation burden in our studied samples. Copy number variants or structural variants in the genome where a DNA segment is present in a different number of copies compared to a reference genome. For example, the regions which are highlighted in red through circles, these samples have a gain in that region when compared to with their normal samples. And the ones which are highlighted in blue, they have a loss of a copy of genome in that region when compared with their normal samples. We also generated the CNV 1 million plot on Data Studio on Cancer Genomics Cloud and found 19Q, 2Q, and 3Q and 2P region to have gain in copy, and while 11Q, 14Q, 16Q to have loss in the region, loss in a copy of the genome. This is an example of how the CNV 1 million plot looks like. This is an example of a patient with the ID 1, and you can clearly see this on the left. There is a new normal screenshot of the CNV 1 million plot from normal sample, and this is from the tumor sample on the right side. And when you compare the tumor CNV 1 million plot from the normal, you can clearly see that there is a chromosome 6q loss. There is a loss in the middle region of chromosome 11q. There's a loss in the chromosome 15q. When we come here, there is a loss at the chromosome, there is a gain at chromosome 8q. Here there is a loss in chromosome 14q, and again a gain of chromosome 19q. We also rule out the single nucleotide polymorphisms, the mutations in our samples. Mutation is a change in the DNA sequence of an organism, and these changes can be caused by errors during DNA replication, exposure to mutagens like radiations or chemicals, or even with viral infections. Mutations can be of two types: germline mutations and somatic mutations. Germline mutations are found in the entire body as they are inherited from the parent, and they can predispose carriers to a higher risk of cancers. Whereas somatic mutations are only found in the affected area, and they arise due to environmental changes or errors in DNA copies. They can provide actionable target for therapy. In our study, we have ruled out the somatic driver mutations. We have filtered the germline mutations, filtered out the germline mutations, and we found these 21 genes to have somatic driver mutations. These mutations were shortlisted as per the list of 299 cancer genes, pathogenicity criteria, and frequency of less than 0.1%. And these were annotated through several databases like Warsum, Nomad, and Cosmic to rule out the true somatic driver mutations. We found that the mutations, we found that two patients have the mutations in PIK3CA, while patient with the ID10 has 7 mutations, somatic mutations. We also found that the TGFBR2 like 3 patients have this TGFBR2 mutation. So PIK3CA is a gene that encodes a protein P110 alpha, which is a subunit of an enzyme called PI3K, and which has a great role in growth, cell growth, survival, and migration. And mutations in this gene can lead to uncontrolled cell growth. TGFBR2 is the transforming growth factor receptor 2, and this gene also plays a role in cell growth and division. And mutation in this gene can disrupt its function because it acts like a tumor suppressor gene, contributing to the formation of cancer. Even this is an example of somatic driver mutation in PIK3CA gene in one patient. And we can clearly see that there is no change here, but in the normal sample, but in the tumor sample, there is a change from G2A nucleotide, showing that this is a heterozygous mutation. Another example of somatic driver mutation in FGFR3 gene, showing that there's no change over here, but there is a change showing homozygous mutation. We also compared the frequency of mutated genes which were found in our sample with the publicly available databases like AACR-Gene and TCGA. And we found that in our samples, PIK3CA and TGFBR2 was the most frequently mutated gene. But in AACR-Gene, we found PIK3CA gene to be highly mutated, but not TGFBR2. When we compared our lab data with the TCGA head and neck positive samples, we found that in this also PIK3CA is the most is highly mutated, but TGFR doesn't have. TGFR is PIK3CA and TGFBR2 both are highly mutated, but not PAX5, but we also see PAX5 highly mutated in our samples. We also saw the enrichment pathways for these 21 mutated genes, and this is the bar graph representing the enrichment terms across 21 mutated genes. And we found that all our 21 mutated genes, they enrich cancer pathways. They are responsible for negative regulation of cell population proliferation, and they come in head and neck squamous cell carcinoma. We wanted to look for the therapies which are ongoing in the clinical trials for any of the mutated genes which we found in our sample, and we found that there is only one drug, which is alpelisib, which is given as a PIK3CA inhibitor, and several trials are, like 31 trials are going on, out of which 21 are open and 10 are closed. Breast carcinoma, solid tumor, and head and neck squamous cell carcinoma are being investigated. However, no clinical trials for TGFBR2, BRCA2, and PAX5 in OPSCC or any other HPV positive cancers are presently in trial stage. This is one thing we should be noted for future, like there should be more targeted therapy options for HPV positive cancers. Coming on to structural variants, these are large-scale genomic alterations that involve segments of DNA, which are typically at least 50 base pairs in length. They encompass a diverse range of genetic changes like deletion, duplication, inversion, insertion, and translocation. Deletion is when you lose a part of the chromosome. Duplication, when a part of the chromosome gets duplicated. Inversion is when there's a change in the orientation. Insertion is when a region of the chromosome gets inserted into another chromosome. Chromosome 4 and 20 here represents the chromosome number. Translocation is when a part of the chromosome gets, like there's a genetic exchange transfer. A part of the chromosome goes to another chromosome, and that part from the another chromosome goes to the first chromosome. So we found so we filtered these germline somatic variants, and this is the table which shows the frequency of somatic structural variants across our samples. And we found that the most of the samples have insertion and deletion variants. Translocations and duplications and inversions were very less. However, in one patient, we couldn't find any somatic structure variant, maybe because the coverage for this sample was very low. This is an example of a somatic structural variant, which was seen in patient with ID5. So there was inversion on chromosome 13 from position 7311638 to 7322646, and these are the screenshots taken from IGV, which clearly shows that in normal we do not find any break endpoints, like no soft clip reads are here, but in tumor we can see that there is the soft clip reads are there, and there is a break endpoint over here in both the positions. So this is so whatever somatic structural variant we found through epitomy workflow, we annotated it through Annote SP tool on CGC, and this is the table showing possible pathogenic somatic structural variants identified post annotation. So in Annote SV, so the annotation is through ACMG ranking of possible pathogenic, benign, unclassified. So I selected those structural variants which are found to be pathogenic or likely pathogenic, and found that these and shortlisted them. These I haven't validated these somatic structural variants through IGV, and presently I am doing that. But the three which are highlighted in purple and green. Purple is the tumor, the genes which are highlighted in purple, like ATM and CSMD1. These are the tumor suppressor genes, and they are found to be muted in several cancers. MUC1 is a MUC1 also plays a significant role in cancer development and progression. However, the rows which are highlighted in red font, I found they have the same structural variant, but I haven't validated through screenshots. So this is still undergoing. So therefore, I would like to conclude my study by saying that episomal HPV-16 is more prevalent than the integrated form in OPSCC cases in our study, maybe indicating that episomal HPV may play a larger than expected role in tumor development. Presently, only PIK3CA inhibitors are in clinical trial for OPSCC. However, other immunotherapies are in clinical trials for HPV plus cancers. So therefore, there is a need for more expanded targeted treatment options. My further works involves identification of ApoBec mutational signatures and identification of differentially methylated regions. ApoBec mutational signatures, they are a specific pattern of DNA mutations which are characterized by abundance of C2T and C2G mutations, particularly in the trinucleotide motif TCW, where W represents A or T. ApoBec enzymes, they are a part of the body's defense against viruses, particularly retroviruses and HPVs, and they work by deaminating cytosines bases into DNA, converting them to uracil. Therefore, looking these signatures would rule out the ApoBec signatures, which can be found in these cases. Thank you. And I would like to acknowledge my mentor, Dr. Michael Dean, for helping me to carry out this work, for his unconditional support and guidance, to all my lab members, and Daria from University of Maryland for providing the samples, tissue samples, and the clinical pathological details, Veera for creating a CNV 1 million plot for me, all the people from Oxford Nanopore Technology for providing bioinformatic analysis support and their technical guidance, and Sarah, Rowan, David from Center Cancer Genomics Cloud for their technical support and giving me a platform to share my work. Thank you, everyone.